Assalamu alaikum wa rahmatullahi wa barakatuh. Mr. Abdullah Amin is talking, and welcome to our biology lesson for grade 9. Today, inshallah, we're going to explain a lesson about the structure of the DNA in chapter 8.2. In this lesson, inshallah, we are going to know the structure of the DNA and how was DNA discovered and what do we mean by base pairing rule. Okay, what is DNA? As we know, guys, that all living things all over the world, even they are prokaryotes or eukaryotes, all of them they have the DNA, and we know that the DNA, which is the genetic material in the cell for all organisms, and the DNA stand for deoxyribonucleic acid. D stand for deoxyribo, and N stand for nucleic, and A stand for acid. As you can see, guys, as you know that the DNA, which is the longest strand, okay, so which is a large molecule, this large molecule is made up of smaller molecule or smaller repeating molecules called nucleotide. And this nucleotide basically, guys, is made up of one here, guys, like a phosphate group, and we have sugar and nitrogen containing base. So the building block of the DNA, which is nucleotide, is made up of three things: a phosphate group and deoxyribose sugar, and finally nitrogen containing base. And the DNA, guys, it has billions and billions of these nitrogen bases along the DNA. So, guys, we can see that. So the structure of the DNA, as you can see, it's made up of nucleotides beside each other. These nucleotides is made up of sugar, phosphate, and nitrogen base, and so on. Sugar, phosphate, and nitrogen base. S stands for sugar and P stands for phosphate. Along the DNA, we have all of these nucleotides. And as you can see, guys, in this longest strand of DNA, we have many kind of bases. We have A, T, and we have C, G. Along this strand, you can find these letters. So what do these letters stand for, guys? So these letters stand for the bases, guys, or the nitrogen containing base. We have A stand for adenine, and T stand for thymine. So adenine always pair with thymine, and we have guanine and cytosine. So as we know that we have four bases of the nitrogen containing base, so we have four types of nucleotides. We have nucleotides that containing adenine, and we have nucleotide that containing thymine and guanine and cytosine. You can see that, guys, we have the adenine can fit by the shape or by the structure of the adenine, can fit to the thymine, and guanine can fit to the cytosine. Okay. We have here, guys, as we said, that the T stand for thymine and C stand for cytosine. A stand for adenine and G stand for guanine. And these bases, guys, as you can see, maybe it's made up of just only single ring like thymine and cytosine, or maybe made up of double ring like adenine and guanine. We call this group that containing double ring purines, and the group that containing single ring, which is pyrimidine. Okay, so we have one pyrimidine can fit to one purine. So adenine can fit to thymine. So the question is, guys, how did the scientists discover all of this? How was the DNA discovered? Actually, guys, the discovery of the DNA is not happening overnight. So it takes a lot of years, and a lot of scientists from all over the world, they contributing to the understanding the structure of the DNA. We have some scientists discovered the chemical of the DNA. Others discovered how these chemicals can fit together. Other guys discovered how the structure or the shape or the three-dimensional shape of the model of of DNA molecule. Here, guys, we have three the most popular scientists which is contributed to the structure of the DNA. We have James Watson and Francis Crick. James Watson and Francis Crick. They like create the structure or the shape, the model of the DNA by using simple tools from the wood and metal. We have also like Rosalind Franklin. She took X-ray image for the DNA, and we have one of the most important scientists, which is Erwin Chorgaff. He found that the amount of adenine equal to the amount of thymine, and the amount of cytosine equal to the amount of guanine, and he named this rule as a Chorgaff rule. Here, guys, Watson and Crick. So they made a model that show the three-dimensional shape of the DNA, and from outside we have the backbone of the DNA, which is made up of phosphate and sugar, and the basis, which is from inside, which made up of the basis that we know, adenine, thymine, cytosine, and guanine. So they make this discovery according to the work, according to, or they built on the work on of Rosalind Franklin, which took an image, and Erwin Chorgaff, which made the rule of Chorgaff. Here, Rosalind Franklin. So the image of X-ray that she took suggested that the DNA was a double helix of even width, and Chorgaff rule state that A equal T and C equal G. The amount of adenine equal to the amount of thymine, and the amount of cytosine equal to the amount of guanine. Okay, guys, so we know that the structure of the DNA from outside and from inside. And we have the bases from the inside and the backbone, which is made up of sugar and phosphate. These bases, guys, is like attached together by certain amount. Because we have always, guys, we have A pair with T and C pair with G, and we name this, guys, as a base pairing rule. Okay, so this base pairing rule show how nucleotides always pair up in the DNA. So we have GC and AT. Okay, guys, so we have like the pyrimidine, which is a single ring, always pair with a purine, which is a double ring, and the helix has a uniform width. One last thing to mention, which is talking about the structure of the DNA also. This backbone, which is made up of sugar phosphate, it's connected together by covalent bonds. But finally here, guys, the bases, nitrogen bases, or molecule nitrogen containing base, it's connected by hydrogen bond. And this is everything about the structure of DNA. I hope you enjoyed this lesson, and inshallah see you next time.